So, dear students, yesterday I I made some videos with regard to color blindness and hemophilia. You know how it passes from one generation to another generation. That is crisscross inheritance or zigzag inheritance or X-linked inheritance, or it is also called sex-linked inheritance. So, there are some stories, interesting stories, but in video I can't put all those things. Let us discuss, if possible, in the month of January or February if college starts, while telling synopsis. Once again, when we are doing crash course of NEET and JEE, I will tell that. But now, in this situation, I have to concentrate only on the topics. So now, let us move to sickle cell anemia. Sickle cell anemia. This is an example of point mutation. This is a best example for point mutation. But you should know, what is point mutation? What is point mutation? See, point mutation is that, for example, this is the first chromosome. Imagine, this is the first chromosome of human being. Is it clear? First chromosome. This is the first chromosome. And you know, inside that, the genetic material will be there. DNA will be there. Genetic material will be there. Is it clear? Yeah. And in that, adenine, guanine, thymine, cytosine, adenine, so thymine, cytosine, adenine, guanine, and thymine. Suppose imagine this arrangement of genes. I think it is clear where it is clear to you whether you are yeah. Otherwise, I will write in a little bit broader manner, okay? Adenine, thymine, guanine, cytosine, guanine, thymine, adenine, guanine, cytosine. Suppose imagine it is going to produce a protein called, that is called, what is that protein? I'll consider melanin, skin color. Melanin. Okay? Suppose imagine we can't get melanin by only 1, 2, 3, 4, 5 nitrogen bases. This is an example I am giving. So, melanin, if it produces, suppose imagine during the time of production of gametes, this instead of G, if A comes here, such there is a change at one particular point. So, this type of changes is called point mutation. This type of change is called point mutation. If there is a change in one base pair, then such a type of mutations are called point mutation. So, sickle cell anemia is such a type of disease. This disease arises due to the change in one nitrogen base sequence. So, dear students, I will explain what is sickle cell anemia now. Okay? This is Autosome linked recessive disease. Autosome, you write the notes. Autosome linked recessive disease. It's a autosome linked recessive disease. It is transferred from parents to offspring when both the parents are carriers. When both the parents are carriers. Say this, we are going to do it. A child will get the disease when both the parents are carriers. Now I should tell what is sickle cell anemia. See, you know that our RBC is dumbbell shaped, isn't it? Our RBC is dumbbell shaped or biconcave. In case of, this is normal, then only if it is in this shape, it is then only it is able to carry the oxygen. From lateral view, side view I'm telling. If when the person becomes a sickle cell or anemia patient, the RBC will turn this shape, my dear students. RBC will turn this shape, sickle cell. RBC will take sickle cell shape. When it takes this shape, then it is not able to carry oxygen. It is not able to carry oxygen. That you should remember. But suppose imagine I am a sickle cell patient. From by birth, I don't know, and my parents will also will not get the awareness about the disease which I am having. Suppose if I am having sickle cell, then imagine. But when I become an age-old boy of 15 years, one day I want to go to a trekking with my friends, or with the NCC or scout from school. Then, while going to the higher altitude, suddenly the persons will get vomiting, giddiness, then the nail bed will become blue, and the eyelashes will become blue, so that and also the lips will become blue, turn blue. And if he is not taken care properly, he will die. So, because when the partial pressure of oxygen is low, automatically the RBC, which is biconcave RBC, it will take sickle shape and it will bend like this. When all the RBCs which are there in the blood vessel, they take a sickle shape, then it will not be able to carry oxygen. It will not be able to carry oxygen. This point you should remember. Is it clear? Yeah. So, now let us see the consequences. What happens? The disease is controlled by a pair of allele, HBA and HBB. The disease is controlled by HBA and HBB. Sorry, HBS. If a person is having the genotype HBA, HBA, if a person is having the genotype HBA, HBA, the person will be normal. The person will be normal. Is it okay? But if the genotype is HBA and HBS, then the person will be normal, but he will be carrier. Person will be normal, but he will be carrier. And when the person becomes HBS and HBS, for both the alleles, he is having a mutated gene, then he will be sickle cell patient. He will be sickle cell patient. So, my dear students, you should remember this particular point, that the hemoglobin, when it becomes HBA, HBA, the person will become normal. And when the hemoglobin is HBA, HBS, then the person will become normal but carrier. And when it is HBS, HBS, then the person will become sickle shaped. Sickle shaped. Is it clear? So this is with regard to the genotype. They say that the disease is transferred when both the parents are carriers. When both the parents are carriers. That means both father and mother, they must have one mutated allele. That means the genotype of them is HBA and HBS. I hope it is clear to you. The carrier genotype. If both are having this, then what happens? I will show. Carrier father with the genotype HBA, HBS, and carrier mother HBA, HBS. HBA, HBS. Then what happens? This HBA and HBS gametes will isolate during the time of gamete formation. Here also HBA and HBS will separate, and then this and this will combine. HBA will combine, and HB normal, I write normal. Then HBA, HBS, the person will become normal but carrier. Normal but carrier. Normal but carrier. Then HBS, HBA. Normal but carrier. Then HBS, HBS. Will be affected. Sickle cell. So what they are saying is correct. Now you can understand. When both the parents are carriers, see, father is also carrier, mother is also carrier. When both the parents are carrier, there is a possibility of getting sickle cell child. There is a possibility of getting sickle celled child. So this is with regard to the condition, how it flows, that disease. Now let us see the reason for this disease. Reason. Why it happens? How it happens? Or what is the reason? The defect is caused due to the substitution of glutamic acid by valine. The disease is caused due to substitution of glutamic acid by valine. I will write full form. Okay? Valine. Val. Dear students, when glutamic acid is there in the gene which controls the structure of RBC, then the RBC will be normal. But when the glutamic acid is replaced by valine, then the person's RBC will not have that firm shape. When the partial pressure of oxygen become decreased, the shape of the RBC turns sickle shape. The shape of the RBC turns sickle shape. Is it clear? So, this is actually the reason. So, at what position of the protein chain? At what position? That is also there. It occurs in the sixth position of beta globin chain. Beta globin chain. In the sixth position of beta globin chain. In the sixth position of beta globin chain. So, because you know, hemoglobin has two chains. One is called alpha chain, and the other one is two alpha chains and two beta chains. Hemoglobin is made up of two alpha chains and two beta chains. So, I will write the structure of hemoglobin. Then you will get an idea. Rough structure I will write. Is it clear? So, dear students, the hemoglobin will be like this. There will be central iron. Central iron will be there. And it is followed by globin. Globin. Here also it will be there. Globin. Globin. And imagine that this globin protein is called alpha chain here, and there will be two beta chain. There will be two alpha chains and two beta chains. Is it clear? Problem will not be there in the alpha chain. Alpha chain will be normal. But the problem occurs in the beta chain. Problem occurs in the beta chain. Problem occurs in the beta chain. It will not be there in the alpha chain. That you should remember. Alpha chain will be normal, but problem will be in beta chain of hemoglobin. That you should understand. Is it clear? Now, I'll move to the structure, how exactly, which is given in your NCERT textbook. So, I'll write as it is. CTC, then GAG, normal hemoglobin. This is the sequence of base pairs for normal hemoglobin. So, it will become, so this is the one which is, this is in DNA. Now, the CTC will produce an mRNA, CG, this is template strand. So this is non-coding strand. So G and T, A, G, mRNA. So this is able to produce an amino acid called glutamic acid. What is that? Glutamic acid. So, 7th amino acid of globin chain. Which amino acid? 7th amino acid. The remaining are same. 6th, then 5th, then 4th, 3rd, 2, 1. 2, 1 are same. No problem. There is no change. And 8th one is also no change. But in sickle cell case, sickle cell, I will call it as HbA peptide chain, HbA chain, because it is normal chain, HbA chain. In sickle cell, CTC will change. Now see, this is one point where the change occurs. Instead of T, here, what the other alphabet that will come? Instead of T, A will come here. Instead of T, A will come here. Then what happens? G, U, G. Now, transcription, mRNA, because this is the template strand. So, instead of C, G, A here, U, and C here, G. G, U, G will come. Then what happens? This will produce glutamic, instead of glutamic acid, what it will produce? Valine in the 6th position. Sorry, in the, 1, 2, 3, 4, 5, 6. This is the 7th. Here, this is the 6th. And 5 are same. Remaining all are same. 3, 2, 1 and 7. So my dear students, all others are same except the 6th position. So now I will call it as HBS chain. HBS chain. Is it clear? Is it clear? Now I will call it as HBS chain. So these are all HBA chain and this is HBS chain. Now let us move to the next aspect. So what is the point mutation occurred here? mRNA now started coding. First, normal mRNA started coding as GAG. Now it has been started coding like, so mRNA started coding GUG instead of GAG. So mutation occurred at one particular point on its template strand, not in mRNA. Mutation not occurred in mRNA. Mutation occurred in template strand. This is DNA. This is DNA. This is also DNA. So this is the template and this is the template. So mutation occurred here that resulted in mRNA and that resulted in the protein. That resulted in the protein. So this is with regard to sickle cell anemia. Now next is phenylketonuria. 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 This is another disorder, an inborn error of metabolism. It is an inborn error of metabolism. It's an inborn error of metabolism. Then what happens? Caused due to autosomal recessive trait. Autosomal recessive trait. So it is an inborn error of metabolism. Phenylketonuria is an inborn error in metabolism. It is caused due to autosomal recessive character. By birth itself it occurs. And it is due to mutation in the gene that codes for an enzyme. Yeah. It is due to mutation. You write down this because it is given in very simple way. If you refer to NCERT, you might be a bit confused. But this is according to NCERT, simplified version. So if you write down this, it is very easy to remember in the exam also, to remember for the exam purpose. So all the points, point by point, you can write. So it is mutation of gene that codes for the enzyme phenylalanine hydroxylase. Phenylalanine hydroxylase. Phenylalanine hydroxylase. What is the function of this enzyme? This enzyme helps to convert, what is that? Phenylalanine into tyrosine. That is the process that should occur normally in human body. What this enzyme will do? Phenylalanine hydroxylase, it is able to convert phenylalanine into tyrosine. It is able to convert phenylalanine into tyrosine. But suppose if that enzyme is not produced, then what happens? Phenylalanine will not be able to convert itself into tyrosine. Phenylalanine is not able to convert itself into tyrosine. So, this affected individual lacks the enzyme. As a result, the phenylalanine accumulates in the cells. In mutated person, phenylalanine starts accumulating in cells as phenylpyruvic acid. Phenylpyruvic acid. It will start accumulating in the form of phenylpyruvic acid. They accumulate in brain, results in the mental retardation. If this phenylpyruvic acid, if it is accumulated in the brain, in other part of the body, slowly body may change its form to some other form. But imagine if it accumulates in the brain, the persons behave like mentally abnormal. The person behave like mentally abnormal. So what happens, my dear students? Phenylpyruvic acid starts phenyl